So hello everybody and welcome back to my YouTube channel and आज के वीडियो में we are going to learn about product development guide. So जब भी कोई प्रोडक्ट नया आता है राइट एक प्रोसेस ऑफ ड्रग डेवलपमेंट ड्रग डिस्कवरी तो ठीक है लेकिन उसके associated simultaneously how many steps are going on right from pre-formulation to a drug approval. तो पहले एक NCE होती है न्यू केमिकल न्यू मॉलिक्यूलर एंटिटी, वो जाती है IND इन्वेस्टिगेशनल न्यू ड्रग एंड वो बनता है हमारा न्यू ड्रग एप्लीकेशन NDA. सो ये जो जर्नी है इट इज क्वाइट लॉन्ग एंड देयर आर लॉट ऑफ स्टेप्स दैट आर इन्वॉल्व, लॉट ऑफ स्टेजेस. सो आज हम वही डिस्कस करने वाले हैं, गाइडलाइंस फॉर डेवलपमेंट ऑफ ANDA प्रोडक्ट फॉर द US मार्केट. सो हम अगर सॉलिड स्टेट्स की बात करें, सो टैबलेट दिस इज द मोस्ट कॉमनली मेड प्रोडक्ट एज इट इज पेशेंट कंप्लायंट एंड लॉट ऑफ डिफरेंट यूजेस. सो सबसे पहले जब आप प्रोडक्ट डेवलपमेंट कर रहे हैं, द फर्स्ट स्टेज इज डेवलपमेंट स्टेज, राइट? And in that stage innovation, you need to learn about the patent existing. So CDR, Central Drug Evaluation Research, Central for Drug Evaluation Research, that is you can search different patents on Google patents or different websites, US patents and all, USPTO, IPO, Indian Patent Office, US Patent Office, right? So you'll come to know whether there are any existing patents. You can search about the literature in Martindale, Merck's, Flore, USP, BP, European Pharmacopoeia. You can search about the different types of formulations and simultaneously. Then you can get electronic databases that are available like Academia, ResearchGate and all. So Mendeley, where you can find articles, and if you want to see about research or dissertations, you can check on shodhganga.com, right? Next up, we have active sourcing. So for active sourcing, we need certain potential suppliers list. मतलब आपका रॉ मटेरियल है तो इट शुड बी आइदर गिफ्ट सैंपल होगा या तो कहीं से आप प्रोक्योर करोगे. सो दोज गिफ्ट सैंपल्स कहाँ से मंगवाओगे? फ्रॉम XYZ कंपनी, सो दैट यू नीड टू नो, राइट? एंड देन सोर्स फॉर APIs. सो इट कैन बी यूरोपियन, एशियन और मतलब बाहर से भी आ सकता है, कहीं से भी आ सकता है API. सो स्टोरेज, ट्रांसपोर्टेशन, so you need to see catalogs, uska catalogs aate, right? So that is very important. The next is the active evaluation. So in this you need to check the DMF availability, which is drug master file, compliance with the monographs. Then you need to know about the impurity profiling, stability studies, right? So according to ICH quality guidelines, there is impurity profiling that is involved. Alag alag guidelines like Q1 is for stability, Q2 is for analytical validation, Q3 is for impurities and all. So ऐसे अलग अलग क्यूरिटी गाइडलाइंस को आपको देखना होगा. Then potential polymorphic forms. So अलग अलग polymorphic forms होते हैं, enantiotropic form, monotropic, मतलब कुछ रिवर्सिबल होते हैं, कुछ irreversible होते हैं, right? Then physical specifications जैसे log P, permeability which is solubility, है ना, crystal lattice, hydrates है, solvates है, what is it, right? Those all things. And then finally the fourth इस परचेसिंग स्टेज, तो आपने अपने सप्लायर को ऑर्डर प्लेस कर दिया. नेक्स्ट एक्टिव टेस्टिंग. सो ऑब्वियसली आप एक COA चेक करेंगे जब आएगा सर्टिफिकेट ऑफ एनालिसिस के द थिंग्स दैट यू हैव, जो आपने ऑर्डर किया है, यू हैव रिसीव्ड दैट ओनली. सो अकॉर्डिंग टू मोनोग्राफ यू कैन चेक और फार्माकोपियास यू कैन जस्ट कंपेयर. Next up, innovator product purchasing. So product purchasing can occur in 3 different stages from large packet size for each product strength. Moving on, we have seventh step, which is product testing phase. So when we talk about tablets, so hardness, dissolution, disintegration, friability, weight variation, uniformity of weight, uniformity of content, ये सब चीजें आप देखते हो. And when we talk about powders, so powder की अलग characteristics होती है, right? We have fundamental and derived properties of powder: tap density, bulk density, angle of repose, Hausner's ratio, Carr's index, LOD, which is loss on drying and all. Then particle size. So all these things you see. And when so that you can evaluate the punch size, right? तो आपका अलग अलग tablet punch machines होती है, rotary die press that you call. So punch size अलग अलग होती है, इसमें die cavity की अलग अलग size होती है depending upon the type of and size of the tablet you want to be punched. Next up, microscopic observation. So again, crystal labate, crystal lattice, particle size, any excipient interactions. You can also check FTIR spectra for drug polymer interactions and all, right? So these are all different techniques that you should employ while the drug is in the development stage. Next is the bulk active testing. So for this you can go for microscopic observation again, polymorphism. So आपका batch approve हुआ, after your batch is approved from the supplier, So full physical characterization and full chemical characterization होता है. So characterization is of three types: physical, chemical, and biological. Biological meat is microbial and proper biological. So you need to do different kinds of testing like optical rotation testing, degradants. So number of degradants, accelerated stability studies, stress condition testing, assays, so that आपको percent purity पता चले. Moving on, excipients. So excipient compatibilities can also be done using DSC, which is differential scanning calorimetry, and you can check the stability of those excipients as well. Then container closure system, which is CCS. So a type of container closure system is very important. आप plastic use कर रहे हो, thermoplastic, आलू packaging, blister packaging, which kind of packaging, and is your product compatible with the packaging? So DMF, when you're filing a dossier, you need to give the CCS, and also CMC is also very important, which is chemistry manufacturing controls. So ये कुछ terminology that you need to be acquainted to when you're when the process of drug development, right? Next is the manufacturing process. So manufacturing process में is, are you going for dry mixing, slugging, chilsonator, wet mixing, order of mixing, आप कब कौन सा, when you are adding a diluent, when you are adding a binder, when you are adding a glidant, then fluid addition when you are adding a solvent, choppers, drying parameters, ये सारी चीजें you need to know, right? Then granulation. So in granulation, particle size distribution, density, flow properties of a powder, compression studies. So in compression, consolidation ratio, weight, thickness, all these things. Stage 12, which is bulk active. So order of active ingredient for PQ. There are different types of qualification: IQ, OQ, DQ, PQ. Installation qualification, operational qualification, design qualification, and performance qualification. So आपके equipments के जैसे calibration होता है pH meter का, वैसे ही there is qualification of equipments, processes for appropriate validation. So आप अलग अलग स्केल पे अपने प्रोडक्ट को, पहले लैब स्केल, फिर पायलट स्केल, फिर पायलट, then pivotal stage, and for pivotal say commercial batch. So this is called scale up, scaling up your product. Similarly, there is scale down. So if there is anything that is missing, then you can scale down. So that is retrospective. So there is just like there is pre-formulation and deformulation studies, engineering and reverse engineering, forward engineering and reverse engineering. So these are a few terminologies that you must know when you are associated with work, especially with the pharmaceutical scientist, right? Next up, we have scope for product development, which is SPC, SPD. So scope for product development is a very specific to the USP medium, especially जैसे dissolution. So dissolution कितने medium पे sorry कौन से medium पे कर रहे हो? Is it a biorelevant medium BRM, or what is the temperature? What is the equipment, paddle, basket, paddle over disc, flow through cell, right? So all these things. So when we talk about this thing, tablets, capsule, of course it is paddle, basket. So how many RPM, 50 RPM, 100 RPM, how many minutes? So all these characteristics you should be knowing. Next is optimization. So optimization for hardness, dissolution, disintegration, friability, all of these things are done, right? And if there are any tablet defects like mottling, lamination, chipping, um, orange peel effect, or anything, sticking and all, तो आप वो सब चीजें को you can optimize and reduce them. Blending time, lubricant concentration, and all of these things are a part of optimization process. Next is step 15, which is analytical validation. So there is IVIVC, in vivo in vitro correlation. So for IVIVC there are three levels: level A, B, C. And there is D also. So one is a point-to-point correlation, one is a single point correlation. So you take Cmax, Tmax, AUC, all the PK parameters for this, and you establish a relationship for the dissolution, right? So there is a single point profile and there is a multi-point profile. So it depends that what kind of profile you are opting for. And finally the scale up. So scale up report is very important for the drug development process, because anticipate कर सकते हैं आप कि आपको कहाँ problem आ सकती है. So you can minimize those erratic variables, those confounding variables in your process. Next is step 17, which is process qualification. So in this you check whether the size of the pivotal batch is appropriate or not. The MFR, master formula record, and BMR, batch manufacturing record, and all is appropriate. The formula is discussed, right? If there is anything to be changed in the formulation process. The next step, we have process qualification again with respect to quality assurance, quality control, RA. So your PQ protocols are very important, and it helps in the overall drug development process. Step 18 is the pivotal stage. सो अब आप पायलट से पाइवोटल स्टेज में आ गए हो. सो अगेन द सेम प्रोसेस, MFR, BMR एंड ऑल, लेबलिंग. बट अभी लेबलिंग, पैकेजिंग, all these things will be added, right, for your particular batch. So this is very important. And finally, since it's a ANDA, so ANDA में सबसे important क्या होता है? BE studies, BAB, bioequivalence studies. So F1, F2, similarity factor, differential factor. So there are particular values of F1, F2, 0 to 50, 50 to 100. How similar or how different is the product? So you need to know. And अलग अलग pH मीडिया में करते हैं काफी बार लोग डोसेज फॉर्म में. 3 different pH मीडिया, जैसे phosphate buffer. Or we usually take 3 different pH phosphate buffer tablets for calibration of pH meter, right? pH 2.6 वगैरह के 6.7 and all. So वैसे ही there are different pH that are used for bioequivalence studies. Finally, the ANDA pre-submission auditing. So in this SOP, you review the SOP, you check the CGMP of manufacturing processes, the protocol is approved, the plant and laboratory documentation are checked whether they are as per ANDA. And finally, the ANDA is filed and 9 copies are submitted like as per the color system, and one is called as the on-field copy, right? And then you submit a ANDA structure in the part of handbook. And validation batches, sometimes they ask to reproduce the validation batches, so 3 consecutive batches are to be kept for inter batch as well as intra batch analysis. And commercial revalidation, so if there is need of SUPAC, which is scale up and post approval changes. So SUPAC has 3 levels: SUPAC 1, SUPAC 2, SUPAC 3. So changes can be mild, moderate or severe, so it depends. So you need to revalidate your process, right? So it's very important that you know about all of this in a drug development process. So that's it from today's video. I hope आपको बहुत कुछ सीखने मिला होगा. And if there's any doubts, feel free to drop it in the comment section, and I hope that you all would have enjoyed the video. Thank you so much for watching the video.